Trypanosoma brucei is a single-celled parasite which can cause the disease known as African sleeping sickness. The parasite is transferred to humans via the bite of the tsetse fly. Once in the human host, T.brucei infects the blood in stage 1, but can then cross the blood-brain barrier in stage 2, causing fatality in cases where the disease is not treated. Currently, most cases of African sleeping sickness occur in the Congo, although due to sustained efforts to control the disease, there has been a large reduction in reported cases for several years now. However, many of the drugs used for treatment are often toxic, and so there is a need for new medications to be developed. This is very challenging, however, since the parasite has a glycoprotein surface coat which protects the underlying plasma membrane. This coat allows evasion from the host immune system due to high genetic variation in the Variant Surface Glycoproteins, or VSGs. These are the most abundant proteins in the surface coat and shield the membrane below from recognition and attack. Embedded within this glycoprotein coat is the transferrin receptor, which is uniquely expressed in the bloodstream form of T.brucei. This receptor is essential to T.brucei since it is the only way for the parasite to obtain iron, required for growth and reproduction. Researchers at the Drug Discovery Unit in Dundee carried out an investigation into the glycosylation of the ESAG6 and ESAG7 subunits of the transferrin receptor, contributing to a better understanding of its structure. We asked Professor Mike Ferguson what led his team to investigate the glycosylation of the transferrin receptor.
Because VSGs usually have either 1 or 2, right? And here, well, per monomer, so 2 or 4. And here's the transferrin receptor, which is related in overall structure with 8. So there's something funny going on. So we thought, well, it's got a lot of glycosylation sites, so presumably glycosylation is important to the transferrin receptor. Let's try and figure out why. So let's first of all find out what the carbohydrate chains are, and then build a model of the transferrin receptor to see how the whole thing looks in total.
On the transferrin receptor, 5 sites of glycosylation were identified on the ESAG6 subunits and 3 sites on the ESAG7, although only 4 sites were positively identified during this research. In this approximate model of the receptor, 6 sites are shown highlighted in orange. Modelling a structure of the transferrin receptor to include these glycosylation sites then gave further insight into the function of these glycans and their role in the transferrin receptor function. Since the VSG proteins are much larger in comparison to the transferrin receptor, it was previously suggested that the receptor could detach from its anchor in order to bind to transferrin and then bring it back to the parasite. With the addition of sugars, however, the receptor becomes wide enough to allow for transferrin to bind without leaving the glycoprotein membrane. This would also not expose the plasma membrane below. The authors concluded that the transferrin receptor has many glycosylation sites to push the VSGs further away, leaving a site wide enough for transferrin to be able to bind without interference. A simple way to visualise this would be to imagine a child in a crowd of adults trying to catch a beach ball. Since they are much shorter in comparison to the adults, it is almost impossible for them to reach the beach ball. However, if they push the adults away from them, they create a large enough space around themselves to allow the beach ball to land on them, similar to transferrin coming in and binding to the transferrin receptor. The glycans have the same effect as pushing the adults, or VSGs, far enough away to allow an interaction. Although this research has not led to any further developments, there remain other possible targets, as Professor Ferguson explains.
So one of the problems with the African trypanosome is that it's got this variant surface glycoprotein coat, the VSG coat, which is quite thick, and almost everything else on the surface is shorter. So the other components are kind of hidden by the VSG coat, and that's why we can't make a vaccine. Although a friend of mine at the Sanger Institute thinks that he's got one for Trypanosoma vivax, which is a related parasite, which I was surprised at, but the data looks fantastic. So, that's why it's always worth continuing to look for things and not just going with dogma that you can't make vaccines against African trypanosomes. So Gavin Wright, his name is, at the Sanger Institute. He hasn't published this yet. Just showed me the data a few months ago. Looks like he may have found a vaccine candidate for that particular trypanosome. But interesting, that's got a much shorter VSG coat. That might be why some of these things stick above the surface.